about him. So he did his PhD from MIT, working with Professor Steve L. Buckwal. You all may aware of Buckwal-Hardwick amination reaction and many phosphine type ligands. So he did 2003 to 2008 is a PhD, and later he moved for postdoc to University of California, Irvine, working with Larry E. Overman. So after that, he took a faculty position in University of Kansas as assistant professor in medicinal chemistry department, and he rose several to associate professor in 2017 up to he stayed in the University of Kansas up to 2020, and after 2020 he moved to Purdue University, and now currently where he's staying. And his main focus research area is organofluorine chemistry and also some medicine application based on the fluorinated molecules. So he has several awards, and he's also serving as the editorial member for ACS Medicinal Chemistry Data as a early career board member, and also Green Synthesis and Catalysis as the editorial board member. So he has several publications. You can visit more for his group webpage. And he's a very good sportsman. You can check his CV. He is a good athlete. So he's a very sportive man. And I had an opportunity to work with him as a postdoc in 2019. It was a very good time, and I learned many things from him. So I hope you will enjoy today's lecture, and I welcome Professor Altman. Now stage is yours.
So hi everybody, welcome. I appreciate you all coming here. I'm going to do a couple of quick adjustments real quick and see if I can make a little bit more visible. So there's one, and then maybe I can slide this over too. And then maybe there's a way to minimize that. Maybe not. Okay, hopefully everybody has a better view of the screen that way. So I appreciate the invitation for hosting me. It's a pleasure to see you again after a couple of years. Ramu was always a quiet individual around the lab, always observing, and I think absorbing absolutely every single detail of everything. And then he would just say one thing that was really funny after not hearing anything from him for days and days. So a very unique character. Good to see you again, and wish you the best. Maybe we should all congratulate him. The group just had a new paper accepted, and he was an author in something that he started in my group, and we just got over the finish line in JOC. So please look at that, another publication on his record. So thank you for your contributions years ago. So the other group of individuals that I do need to thank here, when we click back into the slide deck here. Okay, now the clicker's working, are the students, right? So many of you all in the audience are the most important individuals who make things happen in the group. Fortunately for me, I've been engaged with a variety of exciting young individuals wanting to develop chemistry, wanting to learn, but also produce chemistry. And some of the key players who were involved in this story are shown here. Maybe Brett and Francisco early on, Doug Orsi, who you knew really well. Roma has a co-authored publication with Doug, in addition to some other individuals who are involved in chemistry that I'll show later on in the talk. We obviously appreciate all the funding we get, and the CERB program was nice for funding a student to come work in my lab. So Ankit Patel is currently working with me. He's one of Professor Verma's students from University of Delhi, and he'll go back at the end of the year, I think, and come back here. So it's a great program, and that's what allowed me to come here and engage you all. So hopefully we can have a good exchange of scientific ideas. So the individuals on that slide previously all invested a lot of time and effort in developing organofluorine chemistry. And so organofluorine chemistry for me is really precious. I committed my life and my career to developing organofluorine chemistry. And my interest in organofluorine chemistry go back to graduate school. So I was really amazed at the physical chemical properties of organofluorine compounds, and had the physical chemical perturbations imparted by fluorinated substructures could perturb both the biological properties, physical chemical properties, and change reactivity patterns. So for me, I was really inspired by the physical organic chemistry of fluorine. Okay, and those physical chemical perturbations are obviously important for modulating drug-like properties. And so medicinal chemistry groups are extremely interested in using fluorine and fluorinated substitutes, fluorine substituents for perturbing drug-like properties. That makes it an important need to develop synthetic organofluorine chemistry so that other individuals in the pharmaceutical setting can use these new reactions. So with that in mind, let's see here. Okay, so there's the importance and the need for synthetic organofluorine chemistry. The organofluorine world is a really difficult world in which to work. So synthetic organofluorine chemistry is quite tricky. Historically, there's been many harsh reagents involved, and that has required very unique strategies and unique reactivities. A lot of specialized reagents that we need to be aware of. And in many cases, there are unique opportunities for developing innovative reactions because fluorinated functional groups behave differently than non-fluorinated groups. Okay, so it's a really fun area in which to work. There's a lot of opportunity for synthetic innovation. And in this case, I'll talk about fluorinated alkenes. This is a useful and unique functional group for synthetic organic chemistry. Maybe I can talk briefly about some of the early inspirations in this area. So in the world of organofluorine chemistry, one of the most useful transformations is this deoxyfluorination reaction. There are many reagents that have been developed over decades that can do this transformation, converting simple O-based functional groups into fluorinated variants. Okay, there are a lot of different selectivities and bases you can use, and it's a really tricky area in which to work. And at the outset of our career, my career, I challenged my group to consider converting simple oxygen-based functional groups into higher order perfluoroalkyl derivatives. Okay. If we could accomplish this transformation, it might be useful for a variety of synthetic chemists in applied settings, the pharmaceutical industry, agricultural industry, possibly materials. Okay. Alcohols, ketones, aldehydes are found in a variety of feedstock chemicals, natural products. We can access them synthetically through a variety of transformations. So there's there are many resources available here, and then the ability to convert those into functional higher order perfluoroalkyl derivatives might be extremely useful. Okay. So that was the challenge that I set out with my group to explore. And in this area, we've used two different strategies, decarboxylative coupling reactions and reactions of fluorinated alkenes to try to convert these O-based functional groups into the fluoroalkyl derivatives. If we can learn to harness the fundamental reactivities, we might then move into a period of the group in which we can use these strategies for accessing a wider variety of fluoroalkyl derivatives and fluorinated functional groups, again, for use by the biomedical community. So for those of you who are kind of curious, how would an individual in industry do this deoxy-trifluoromethylation sequence? It's a 4-step synthetic sequence that requires oxidation of the alcohol to a ketone or an aldehyde, and then the use of the Rupert-Prakash reagent to do a net 1,2 addition reaction, generate a trifluoroethyl alcohol, and subsequently take 2 steps to defunctionalize. Okay, a reasonably lengthy sequence with a lot of inefficiencies, and presumably we might be able to improve upon the synthetic sequence. Nonetheless, many useful molecules have been accessed through the synthetic sequence. You can see some nucleosides, amino acid derivatives, other small heterocyclic molecules, and even some sugar mimics. Okay, so it's been used over and over again in the industry. Nonetheless, the goal of the group was to take simple ketones or alcohols in a single step, convert them over to the fluoroalkyl derivatives. And that's how we're going to start this story. Initially, we explored copper-based catalysts that would engage a halo difluoroacetate precursor. So these would decarboxylate, lose CO2, and replace either the chloride or the bromide by fluoride. And in the presence of copper, this would make copper CF3, which is a nucleophilic source of CF3 that could then react with a variety of aliphatic or activated C-based electrophiles. Mm-hmm. Mild conditions, good functional group compatibility. That's something that you can check if you look back in the papers. Now, the fundamental reactivity in this area was established by Professor Chen at the Shanghai Institute of Organic Chemistry some 30 years ago, who took the alcohols, converted them into halodifluoroacetate esters, and subsequently used stoichiometric copper and super stoichiometric quantities of other reagents to convert the halodifluoroacetate esters into the trifluoromethyl derivatives. Just getting a little warm in here with the and humid for a coat, so pardon that. Okay. And at the outset of the group, we sought an opportunity to develop a catalytic variant of this reaction. And there were a couple of key features of the mechanism for Professor Chen that inspired our thinking and encouraged development of catalytic processes. So without walking through the entire mechanism, I might suggest 3 points to focus on. The first is that the reaction started with copper iodide. It generated copper iodide at the end of the reaction. So you envision that you should be able to re-engage that copper iodide and get it to re-enter the catalytic cycle. The second key feature of this reaction is that the first step generated an iodide. And that iodide then again reacted in the very last step. So modulating the concentration of that iodide intermediate might enable us to control these reactions. Okay, so that was the second key feature. And then the third key feature is that this reaction actually was proposed to generate free difluorocarbene and CF3 minus. Right? And those are reactive intermediates. They react with a variety of carbonyls through 1,2 addition or addition-elimination reactions. Okay, they'll deprotonate acidic protons reasonably well. And if this mechanism were operable, it would necessarily limit functional group compatibility. Okay. Nonetheless, we were inspired to develop a process and hypothesize that the decarboxylation, the formation of CF3, might occur at the metal center, which would improve functional group compatibility relative to what was proposed previously. So over the course of the first few years of my group, we developed a series of catalytic reactions that used copper in the presence of a diamine ligand to do a net decarboxylative trifluoromethylation reaction. And this worked with allylic substrates, benzylic substrates, and propargylic substrates. In the final line there, the propargylic substrates could be converted to either the propargyl or the allylyl products in modest to excellent selectivity using a ligand control strategy. Right? So the DMEDA ligand, which was popularized by my previous mentor, delivered the propargyl products, and the phenanthroline-based ligand delivered the allylyl products there. 2 key features to note of this chemistry. In most cases, we use an excess of that halodifluoroacetate. Use the sodium bromodifluoroacetate or the methyl ester in the reaction conditions. And that was important for activating the catalyst, generating a copper CF3 so that it could enter the cycle. So it was a pre-catalytic event. The second key feature is you can see in the line of the benzylic substrates there, we actually add extra I minus in some of these reactions, again, to help modulate the concentration of that reactive intermediate, increase it to facilitate catalysis. Okay, that's not essential for all of the different substrates, but it was important for some of them. And these are reasonably inefficient processes. That's by far not a perfect process. The esters themselves still take a step to prepare. In addition, they're not the most stable. Some of these esters decompose on silica or with prolonged storage. And so we sought to develop an alternate strategy in which we could use a new reagent to do a deoxy trifluoromethylation reaction. Okay, so as opposed to a 2-step sequence, as I just showed you, we decided to develop a new reagent to connect these functional groups. And so, long story short, phenyl bromodifluoroacetate is an ester that promotes the deoxygenative trifluoromethylation. I'll briefly comment on how this reagent works. Initially, there's a condensation reaction between the alcohol and the phenyl bromodifluoroacetate ester to generate the intermediate that we saw previously. Now, this initial step is catalyzed by KF. We've run several control reactions to demonstrate that KF mediates this process. And then in the same pot, the copper-based catalyst system will react to decarboxylate and do the substitution reaction, ultimately delivering the CF3. And this reaction works with a variety of substructures, useful functional groups, further synthetic elaboration, heterocycles. You can see the synamyl other allyl, benzyl, heterobenzyl, propargyl products can all be accessed from this reaction. So that's the copper catalyzed decarboxylated chemistry. It does help enable access to real world molecules. So this is one example of a heterobenzylic alcohol that a group in the agricultural industry wanted to convert into that trifluoromethyl derivative. And that sequence that I had mentioned before takes 4 synthetic steps. The initial oxidation of the alcohol to the aldehyde, 1,2 addition of CF3 minus to deliver the trifluoroethyl alcohol, that's the intermediate shown, and then 2 steps to defunctionalize the molecule, in this case using stoichiometric. In contrast, using the first series of reactions I showed, we can synthetically prepare, isolate, and purify the bromodifluoro intermediate, then subject that to a copper-based catalyst system and deliver the product and double the yield in half the number of steps. In contrast, we can access that same product in a single step and near the same yield, 60% there, using the phenylbromodifluoroacetate reagent. Okay. So that's one of the strategies we've used to accomplish this deoxy-trifluoromethylation reaction. There's a lot of chemistry there that I'm just summarizing because I want to get on to the next topic. Okay. We've used palladium-based catalyst systems for doing decarboxylative coupling reactions to access fluorinated ketones, which are important for medicinal chemistry and chemical biology. But I really want to move on to a fun topic. This is a really fun and exciting topic for me because of the physical organic chemistry involved in this area. And that's the reactions of fluorinated alkenes with a variety of different reagents to generate trifluoromethanes and then more broadly other fluorinated functional groups. Now, to best understand the reactivity of fluorinated alkenes, we might consider other activated alkenes with which many of you are more familiar. Right? For example, there's a variety of electron withdrawing groups that will activate alkenes through a resonance effect. And this activation occurs at the beta position, and a variety of nucleophiles will then attack that beta position. When that reaction occurs, we typically push the electrons up to the alpha position. There are opportunities for further functionalization from that intermediate. And there are a variety of functional groups that one can use to activate the alkene through this mechanism. Now, in contrast, fluorinated alkenes are activated by a different mechanism, and that's an inductive effect, right? The fluorine atoms are sigma withdrawing groups that instead of activating the beta position will activate the alpha position. So nucleophiles would preferentially attack the difluorinated position, which would enable further functionalization at the beta position, right? We push that anion down, maybe you can trap it with something else. Okay, so different mechanisms of activation enable access to different types of products. And to understand, I think, a bit more the distinct reactivity of these fluorinated alkenes relative to other non-fluorinated alkenes, we might consider these fluorinated enolates, right? These fluorinated enolates are actually not nucleophilic at C alpha. Right? Usually when you think about enolates, you think about them being nucleophilic at this position. In contrast, fluorinated enolates are actually electrophilic. They'll react with organometallic reagents, strong nucleophiles, through a substitution process that replaces one of those fluorine atoms with whatever your alkyl group is. And subsequently, you get these substitution products up here. So it's an Oompa type of an alkene there compared to what you're used to. Okay? So this is the goal of showing this was just to demonstrate how the, okay. Now, the fluorinated alkenes can be accessed by a variety of different synthetic methods. There are many strategies for accessing these. Some of the most direct might involve cross-coupling reactions, olefination reactions of carbonyls. You can see halide and boronic acids, diazo compounds can all be converted with different types of catalysts. So there are a variety of strategies for accessing these. So just to demonstrate the reactivity of these intermediates, I'll demonstrate how we've used this strategy for converting ketones and aldehydes into trifluoromethanes in a single pot. So the reagent we use in this case is this phosphonium beta-ene there that decarboxylates under thermal conditions to generate a phosphonium ylide. The ylide reacts with the aldehyde in a Wittig olefination reaction. And then you can see how the fluorinated alkene will react with a nucleophile here at this alpha position. So fluoride can attack in, that generates your CF3 and presents an anion down here that subsequently gets trapped by H2O. And in your standard bottle of TBAF, THF, you usually have about 5% water in those bottles. They're sure sealed, but you still have quite a bit of water in there. So this reaction works to generate a variety of fluorinated substructures. You can see these benzylic trifluoromethanes bearing electron donating withdrawing groups. Heterocycles are tolerated in these cases. Basic nitrogen atoms are tolerated. You can scale these up. And they're metal-free conditions, so they enable functional groups that can further react with cross-coupling conditions there. Okay. In the case of ketones and converting those into secondary trifluoromethanes, we actually use a slightly different reagent. So in the previous case, you saw that Wittig precursor had the triphenylphospheno group attached to it. And here we have the tris diethylamino group there. Okay, so it's a little bit stronger of a nucleophile when it starts that Wittig olefination reaction, but that's needed to convert the ketone-derived substrates into the gem difluoroalkenes there. And these reagents were isolated, purified initially by Jingsheng Chao at the Shanghai Institute of Organic Chemistry. We kind of borrowed their reagent for our purposes. These reactions, too, enable access to a variety of secondary substructures there. You can see some of the examples we have here. Again, useful functional groups for medicinal chemistry or further synthetic elaboration. And this also enables access to target molecules. So this is the same example you saw before. We have an opportunity here to intercept the aldehyde intermediate in their synthetic sequence. Okay, so instead of doing this transformation in 3 steps and 40% yield, we can do it in 1 step and nearly 70% yield. Okay, avoiding transition metals, this occurs in a single pot, about 1 day. We can cut off a couple of steps from a synthetic sequence. Okay? So again, enabling access to target substructures that other individuals. Now, I started off this section suggesting that we might have opportunities for difunctionalization, okay? And we've explored that to some extent in the case of using other nucleophiles. So first, let me demonstrate a problem in this research area, okay? The last reaction involved nucleophilic attack at the alpha position, formation of an anion that we quenched. And if we use other nucleophiles, this actually becomes particularly problematic. So for example, if we take some type of an oxygen-based nucleophile, we do that initial addition reaction, that generates an anionic intermediate that has beta fluoride atoms. And the beta fluorine atoms are actually prone to eliminate and release F-. That's a really good leaving group. And so even though we want to protonate that intermediate, instead there are many more examples in literature of losing fluoride and delivering these monofluorovinyl ethers. Now there are some workaround examples. Maybe I can first demonstrate how easy these reactions are to do. So you can see the conditions there, potassium carbonate, room temp. It doesn't take much to do this additional elimination reaction. Okay, so that beta fluoroanion is really prone to eliminate. There are some workarounds using oxidation and reduction in the same 2 steps to generate the desired CO bond, but that's definitely a suboptimal sequence using an oxidant and then stoichiometric tin in the next step. So some years back, we considered using an organocatalytic base strategy for generating these CF2 heteroatom bonds. And in this proposed mechanism, we're going to use a catalytic weak base to establish an equilibrium there between the nucleophile and the base that would generate low concentrations of the nucleophile that again react, generate your anion that has those beta fluoride atoms that are problematic. And if you can modulate the concentration of the reagents properly, you might be able to protonate that intermediate using the conjugate acid of your base or another equivalent of the nucleophile. Okay, this would be challenging for 2 different reasons here. The first, that if the conditions are too harsh, you might do HF elimination because you're the basic conditions. The second is that you still have to outcompete the unimolecular decomposition that can occur. Okay. And so Doug and Jacob were really important drivers in developing some of the chemistry and helping address this problem. Now, early on, we had a success to some extent with this organocatalytic base system. You can see a couple of examples here. In the case of phenols, TBD served as a suitable base for delivering predominantly the difluorinated ether as opposed to the monofluorovinyl ether. Okay. Formation of CS bonds was a little bit more easy. Sulfur being more nucleophilic might generate the CS bond under more mild conditions, and that was definitely the case in our hands. Lower temperature using lower concentration of the weak base facilitated the formation of the CS bond, and there was much less elimination in these reactions here. Okay. A lot of reactions to develop those systems, a lot of optimization, as you're well aware. Nonetheless, this is what we came to as a conclusion. You can see some of the substructures that are accessible using this method. In general, these are all aryl nucleophiles, so phenols and biophenols. We made attempts to extrapolate this to aliphatic nucleophiles, and I'll comment on that as we move forward and show you some other catalyst systems. But a variety of useful functional groups, target substructures that really aren't represented much in literature were accessible using these strategies. Now, as we did try to move forward and extrapolate these reactions to aliphatic substrates, we faced many challenges. If we use the original reaction conditions, we did not selectively form this difluorinated product. We saw a lot of beta fluoride elimination, ultimately providing the fluorovinyl ether as the major product, bioether, pardon me. There were many attempts at optimization of this reaction, and many times we optimized what we thought was going to be a good reaction, and then ultimately it didn't extrapolate to a broad set of substrates. So here's an example of one of those stages in which we used a catalytic equivalent of the conjugate base of our nucleophile. It seemed like it was working really well. That's an awesome result, right? And then you try to extrapolate it to other substrates. You can see the selectivity dies off as you change the electronic substructure of that arrow ring. Okay. Ultimately, we came to a catalyst system using lithium triflate and pyridine that provided a reasonable scope for reacting difluorinated styrene with these aliphatic thiols. A couple of other key features. We use methoxyethanol as an additive in this reaction. I'm going to come back to this and talk about air, because air actually is really important for this reaction, and I'll explain why in just a minute. First, I'll just demonstrate that this reaction enables access to a reasonable set of substructures. A lot of useful functional groups are tolerated on the styrenyl portion of the molecule. And I talk about styrene because this is difluorinated styrene. Heterocycles are tolerated. You can see over here on the right, we can selectively react a fluorinated alkene in the presence of a Michael acceptor. Other useful sensitive functional groups, alkyl halides, esters, we can selectively react a alkyl thiol in the presence of an alcohol. And that should be expected based on the nucleophilicity of sulfur. But I want to come back and think about mechanism and selectivities. And I want to rethink that hypothesis that organocatalytic bases were driving this reaction. So if you go back to your pKa charts and you check the pKa's of thiols versus the pyridinium, which we might initially think be forming, those pKa's are actually in the wrong direction. So the equilibrium does not lie in the direction we need to generate an anionic thiolate. And this is like 10 to the 14 fold, right? This is a huge, huge, huge value. So that's energetically just totally the wrong way. And this contrasts the previous couple of reactions, right? In the previous couple of reactions that I showed, the pKa's did favor formation of the anionic nucleophile. Okay, so those first two reactions using these catalytic bases, we do think are reacting via the mechanism proposed. But as we went through and developed this final reaction, something's wrong, something's not working right. Okay. And digging through the literature, it became apparent that maybe we're getting to a pyridinium by a distinct mechanism. Okay? And I had mentioned before that air was important for these reactions. And thiols oxidize, right? We know that. So if you look back in this old literature, thiols will react with oxygen through a variety of steps to generate sulfinic acids, and this can further react to make sulfonic acids. And maybe that sulfinic or sulfonic acid might be responsible for reacting with pyridine and generating a pyridinium. And then we have an active catalyst that might be operating by a distinct mechanism. Okay, so we kind of stumbled across this mechanism. We did explore it. I'll walk through the mechanism itself and then talk about some of the mechanistic support for this interesting and unusual mechanism. So initially, there's an activation process that converts the pyridine to a pyridinium. That pyridinium can then react with the fluorinated alkene and a thiol in an ordered transition state in which the pyridinium transfers the proton to the alkene as the nucleophile is attacking. I'll comment on some of the support for that unique mechanism. That would generate the CS bond simultaneously with protonation, so we never make that anionic intermediate. It's avoided in this sequence. And then a late stage proton transfer would generate the product and regenerate the pyridinium. Okay. So let's talk about some of the support for this mechanism. So first, okay. So in the first case, we did observe some of these sulfinic and sulfonic acids by mass spec analysis of reaction mixtures. Okay, we could detect those in a couple of % concentrations. Okay, so that was important. We did that stoichiometrically in the absence of some of the other reagents. In addition, we did it in reaction mixtures. The second key experiment that we ran was to take the independently synthesized pyridinium triflate and use that in the catalyst in the reaction. And that was kinetically competent as a catalyst. Okay, so the kinetics matched near straight on. And we could actually run this reaction in the absence of air, and it worked when we used this catalyst, suggesting that that might be the active catalytic species. We do have support for a transition state that has built up anionic character at this position here. So we observe a linear free energy relationship, and that correlation occurs with sigma minus, right? It's a low value, minus 0.41, and again, that would be consistent with buildup of anionic charge at this position. Additionally, we observe a really strong kinetic isotope effect. So KHKD on the order of 4 to 5 is supportive of a proton transfer being involved in the key step. And again, that proton transfer would be involved here, right? The breakage of the nitrogen-hydrogen bond there versus the nitrogen-deuterium bond is involved in the rate-limiting step of this transformation. Okay. So it's an unusual transition state there. Nonetheless, it might be operable in these reactions. Now, this was still a reasonably limited reaction. All the examples I showed you to date actually bear this fluorinated styrene substructure. Okay, so you need some type of an aryl group here to activate the alkene, and that helps stabilize some of those intermediates that I've been talking about. As we move forward, we've been trying to develop a strategy that will enable us to couple a variety of nucleophiles to aliphatic substructures there that are not similarly activated. And here's an example of that we're getting close to submitting for publication using a photoredox-based catalyst system to generate S-base radicals and add those into the fluorinated alkene and then ultimately quenching the resulting intermediate. And this reaction enables us to get into aliphatic and heterocyclic substructures, such as those shown here. Some basic nitrogen atoms are tolerated. You can see that on the bottom. Other functional groups useful for the synthetic elaboration are also tolerated. Sulfur nucleophile in the presence of a basic amine. So this is getting to a more broad, more generally useful strategy for forming these CS bonds. One key note, and one thing we're ultimately going to need to explore a little bit more, is the role of this additive here. So that's an aniline derivative. A variety of aniline derivatives were really important for facilitating this reaction, possibly serving as a mediator to help generate the appropriate S-base radical. But there's more to come on that. So at the outset of this talk, I mentioned some of the unique properties that fluorine can impart in organic molecules, and you know, we do enjoy studying that also. I love the physical organic chemistry. Here's a series of examples of how fluorine can perturb nucleophilicity and oxidative stability of those thiols that we've been making for several slides now. So if we consider the nucleophilicity of these thioethers, they can react with a variety of alkylating reagents, in this case methyl iodide, to generate sulfoniums. Okay. Here we're just looking at how much of the material is remaining at the end of an hour, or sorry, this is an 18-hour reaction. So if you have the non-fluorinated thioethers, they'll alkylate reasonably easy. You can see about 35% of the material remains at the end of the reaction. The fluorinated thioethers don't get touched by that methyl iodide. Right? So they are really less nucleophilic compared to the non-fluorinated variants. In addition, fluorination of these thioethers stabilizes the molecules relative to their oxidative stability here. So here we're doing an oxidation reaction. We're comparing and match pairs, the non-fluorinated versus the fluorinated variants, and look at how much of the material is remaining at the end of the reaction. Okay. A variety of oxidants, such as some of these reagents, will decompose a substantial quantity of the non-fluorinated thioethers. This is just a 1-hour reaction here. Nonetheless, the difluorinated thioethers are reasonably stable to a variety of oxidants. Only MCPBA will decompose these intermediates. So there's a significant change in the oxidative stability of the thioethers. And for us, this is interesting for medicinal chemistry purposes because oxidative metabolism of drugs is really important. If you can avoid that by introduction of fluorine atoms, that might be useful for developing in vivo probes. Okay. So again, just one example of how fluorination can perturb properties here. So I entered this section talking about this organocatalytic-based strategy for engaging the alkenes. I took a little sidetrack and started talking about some other strategies for reacting to these fluorinated alkenes. But as the group has moved forward, we started to ask the question, well, what if you could trap this intermediate? What if you can get these intermediates to engage with a transition metal or some other oxidant to start doing difunctionalization reactions? Okay, so some years ago, we had this hypothesis that you could generate this anion, trap it with a transition metal, and then with some other oxidant or reagent, generate products of 1,2 difunctionalization. And this was a little bit of a naive hypothesis initially. So such difluorinated beta metallospecies have been made in the literature, and they're actually key intermediates for a variety of reactions that actually undergo beta fluoride elimination also to deliver monofluorovinyl products. Okay. And there are a variety of catalyst systems that have been used to do these defluorinative processes. CH functionalization reactions, cross-coupling reactions. There's actually a whole host of transformations. I'm just highlighting a few of them here. This might be a general review of some of the reactions that do defluorination. Okay? That's definitely not what we wanted. We've been interested in avoiding defluorination. It's so hard to put fluorine onto a molecule. Why would you want to rip it out? Is my view on the world of fluorine chemistry. And so for the last couple of years, we've been exploring strategies for avoiding defluorinated processes. And one of the strategies we've used is to use radicals as key intermediates. So if we can generate nucleophilic radicals that will react to generate this new nucleophile CF2 bond and present a radical at the beta position, these would not undergo beta fluoride elimination, right? We could then engage that radical with some type of oxidant and further allow the transition metal to decompose this intermediate and generate useful products. Okay, so the hypothesis there is that we can use radicals as intermediates. And we've used a couple of different strategies to accomplish this goal. So you can see a cobalt-based catalyst will do a difunctionalization reaction in which we generate a new carbon-oxygen bond here at the difluorinated position and a carbon-oxygen bond here at the benzylic position. Using an alternate copper-based catalyst system, we can generate the same CO bond here at the difluorinated position, but these will further oxidize to generate the ketone products. Okay, so the different transition metals have the ability to access 2 different products. And in these 2 cases, we're using molecular oxygen for forming that second bond. And again, these reactions work for generating a variety of substructures for both reactions. I don't want to spend too much time looking at substrate tables. Nonetheless, you can see that there's near exclusive chemoselectivity for the copper-based system here on the right, a variety of useful functional groups. They can react on natural products, natural product derivatives, such as this dextromethorphan derivative over here. In contrast, the cobalt-based system delivers the alcohols with modest to good selectivity. Okay. And so both of these reactions, without going into too many details, likely proceed through some type of peroxo species and the copper or the cobalt fragment to deliver the products. Now, as we move forward, we've also explored another strategy, and that's by offering alternate low-energy pathways to avoid beta fluoride elimination. So this is an example of a reaction in which we're going to allow a transition metal to bind to the substrate and then encourage the transition metal to eliminate a beta fluoride instead of a beta, eliminate a beta hydride instead of a beta fluoride there. Okay, and if the kinetic competition favors beta hydride elimination, we can avoid the beta fluoride there. Okay. Subsequently, the metal hydride that's initially delivered can do a chain walk. You've seen that plenty of times in your organometallic classes to deliver the thermodynamically stable products. So really briefly, I'll introduce you to the catalyst system that does this transformation. The reaction of these difluorinated alkenes with aryl sulfonyl chlorides will generate the CF2 bond and then walk the alkene to a thermodynamically stable position. It's a palladium-based catalyst system using an NHC ligand, stoichiometric copper. Those will all be relevant as we move forward in the discussion. Okay. You can see a variety of substructures tolerated here. Aryl sulfonyl chlorides, heteroaryl sulfonyl chlorides are useful in a variety of interesting alkene derivatives here. Again, we're not using styrenes because we need to have hydrogen atoms here at this position to allow for that beta hydride elimination to occur. So our mechanistic hypothesis for how this is working currently is shown here. We have a palladium-2 based catalyst system, and we think that this is actually proceeding through that. We don't think it engages in a palladium-02 cycle. The palladium-2 based catalyst system can engage the substrate, and then a combination of reagents are actually required to activate the aryl sulfonyl chloride. So I'll suggest that palladium, copper, and the lithium carbonate are all responsible in this step here. Ultimately, the metal will facilitate the formation of the carbon CF2 bond, generate that reactive intermediate. Here I'm showing a palladium-3. We'll talk about that in a couple of minutes. That can do the beta hydride elimination, generate our metal hydride that can then undergo a reinsertion process and walk that alkene to a thermodynamically preferred position. Okay. Ultimately, there'll be a process for reducing the metal back down to our active palladium-2 form. So I'll show you a variety of experiments. I'll talk a little bit about this activation process. We'll talk about some radical traps and demonstrate what's going on here in this portion of the cycle. And then I'll demonstrate the chain walking using a deuterated substrate and show the scrambling of deuterium as you would expect. So I commented before, a variety of reagents are responsible for activating the aryl sulfonyl chlorides. So here's an experiment in which we take the aryl sulfonyl chloride and just look for dimerization and decomposition of that. If you use all of the reagents, we do get full conversion of the substrate. We see bi-aryl, we see some aryl chloride forming also, right? We're starting with stoichiometric copper chloride there. So that's one of the byproducts is an aryl chloride. If you remove palladium, the substrate doesn't react, it doesn't convert. But also, if you remove either the copper or the lithium carbonate, you see low levels of conversion there, so inefficient activation of the aryl sulfonyl chloride. Okay, so all three of these reagents, we're not quite sure how it works, but all three of them are responsible for activating the substrate somehow. We do have evidence of radical intermediates, so we do trap out intermediates using BHT. So, in this case, we're only subjecting the aryl sulfonyl chloride to BHT under the reaction conditions, and we see two different products, one that would form via loss of SO2 to make an aryl radical and then trapping. In addition, another one in which the SO2 is still attached to the aryl ring and that traps with the BHT. Okay, so presumably there's an SO2 radical in addition to an aryl radical in this mechanism. We observe these same two side products in full reactions, and they are kinetically competent, kinetically competing with formation of the actual product here, right? So, you know, a little over a third of the material here and about a third of the material there. So again, suggestive of aryl radical intermediates there. Now, palladium actually has two important roles in this catalytic transformation. I've already commented a couple of times on the beta hydride elimination, but I also want to suggest that palladium is important for formation of the aryl CF2 bond. So in these reactions, we've had some computational support from Paul Chung and his group. You'll see his picture in just a minute. And we're suggesting a palladium-mediated formation of the aryl CF2 bond, in which you have formation of a metal carbon bond and aryl CF2 bond at the same step simultaneously. Computationally, that's a low energy pathway relative to some type of an anterofacial carbopalladation, which was also calculated, or outer sphere attack, in which an aryl radical just attacks the CF2 position to generate a carbon-based radical here at the beta position. Right? That's extremely high in energy. So palladium is important for that initial step formation of the CC bond. There's additional evidence of palladium playing an important role, again, that beta hydride elimination. We do have experimental support for this step, and that's the scrambling of deuterium. So if we subject this substrate here that bears the two deuteriums there, we see scrambling as you'd expect, and is consistent with some other reactions that involve a chain walking process. Okay? So that's strong support for the beta hydride elimination. So Paul Chung and his grad student Taisia further explored this process and compared kinetically and computationally these palladium hydride and palladium fluoride elimination processes using this model system shown here in addition to some others. And the palladium hydride elimination was always faster than the palladium beta fluoride elimination. And that was studied in a series of different substrates. So in this case, you can see this is a substrate that on one side has a benzyl substituent, on the other side we have a homobenzyl. You get nearly the same results if you flip those two and switch the benzyl and the homobenzyl sides. Okay, so we looked at a variety of substrates here. There are a couple of other key features here. There seemed to be a lot of difference in the distortion energy for beta fluoride elimination relative to beta hydride elimination. So bond cleavage of the CF bond is more difficult than the CH bond. That was demonstrated computationally. The other really interesting feature of this chemistry is that we did explore palladium-2 and palladium-3 processes. You don't see palladium-3 very often. It's really unusual. And I'm not always comfortable talking about palladium-3 because you see it so infrequently. Okay. Nonetheless, their calculations suggested that the palladium-3 was significantly lower in energy than the palladium-2 processes. So again, a little computational evidence to support that hypothesis, even though it is a little bit of a stretch. So, you know, hopefully I've started to encourage you all or excite you all about the uniqueness of organofluorine chemistry. There are a lot of unique reactivities. These fluorinated alkenes embody some of my excitement in the area. They behave really differently compared to other alkenes, and it's a really fun area in which to work. Okay, so yeah, fluorination is tricky. You get a lot of weird intermediates. You got unusual reactivities. You get side products you don't expect. Like, it's a really fun area to work in. We need specialized reagents. There's a lot of opportunity for creativity and new reaction design, new reagent design. And so hopefully, you all get inspired to think creatively about chemistry. Other than that, I appreciate the opportunity again to visit you, to see you. It's a really cool time to be here and see the culture and see different ideas and talk with you all. I look forward to further discussions. I'm happy to engage in a Q&A that you all have.
Thank you, Professor Altman, showing to our chemistry and also recent development, and particularly excited about the palladium 3. So I will discuss it later. But now session is open. You can ask. I have a question regarding how much equivalent you use higher because that is more also.
Yeah. In the thiols, the reactions work typically about 1.5 to 2 equivalents of thiol. Some of the reactions we can get them down to 1.1 equivalents. So it's not like we have 10 equivalents, right? It's usually closer to a stoichiometric quantity.
Because it's oxidized and it dimerized. So if you use one equivalent, so it becomes 0.5 because it's dimerized.
Yes.
Also, going for the acid, thereby generating that this acid, that pyridinium.
Yeah. Yeah, so for let me pull up that system real quick so we can talk about it. So that system, we don't use a major excess. Oops. It's actually my dad's birthday today. So everybody say happy birthday to my dad. Get back to this page. Okay, there. So that was this system here, right? And so this is not a major excess of thiol. So we can do these ones with about 1.5 equivalents. You're right that there is some dimerization of thiols, but the dimerization of thiols doesn't occur greatly, okay? And specifically in this case, we're actually, I think the dimerization of thiols is useful because if they further oxidize, we get to the sulfinic acids that we actually want to generate the active pyridinium. Okay. Now, truth be told here, if our mechanistic understanding changed a lot as we went through the project, right? And so we weren't aware of this oxidation process when we started it. We weren't gambling on it. We just noticed certain reactions were working in the air. And so maybe I can go, that's not the slide I want. Back to this one. If I were to go back in time and we knew then what we know now, we wouldn't do these under air. We would do them under nitrogen and we would just throw in a couple of percent of this and the reaction works just fine. Okay. And we've done that with a series of substrates that didn't get published, but it was just one of those things like, dang, that would have been cooler if we would have done that. But we learned a lot mechanistically about what's going on by the way we develop the chemistry. And that's the way it works sometimes, right? Sometimes you get reactions to work and you figure out why they work or why they're unique later. So yeah, but you're right. We do observe some of those dimers, but if they further oxidize to the reactive acid, to the acids we want, then it's okay.
Slide only you had shown that gauge by KD per hour. Can you please explain that step? I somehow missed that.
Yeah, so the thought there is that in this transition state, the transfer of the proton from the pyridinium to the substrate occurs. So the KHKD that we're observing is this one here. Okay, we're breaking the NH bond. Sorry? It's not an anion. There's anionic charge on it, but it's not an anion. Yeah. So the anionic charge we can observe, right? There's buildup of anionic charge at the transition state. We have an LFER. And the correlation there is actually pretty darn good, 0.92, right? So so maybe whiteboard, chalkboard, I guess it's over there, but we don't have a pen. Okay. So, maybe something to think about here is the nature of the fluorinated alkene itself. Okay. I talked before about sigma inductive effect that generates delta plus at the alpha position, but also think about the hyperconjugative effect. Fluorine has lone pair of electrons, and it's a second row element. So the same energy level, same shell as carbon, right? And so hyperconjugation between fluorine lone pair of electrons and carbon-based orbitals is no, and it's a really important feature. So if the fluorine electrons donate into the pi system, you would increase anionic character at the benzoic position as shown here. And so that position is intrinsically delta minus. And by offering the pyridinium, we can start to protonate, right? So I'd say yeah, that's going to be delta minus. And so that'll protonate.
Like if it is like around 5 means it's a primary kinetic.
Yeah.
State.
Yeah. Yeah. So that's a primary effect. There's a really strong dependence on the rate that's involved in this. Again, the proton is transferring from here. So you're breaking. So think of it not as, I think you're thinking of it as formation of this. I want you to think of it as breaking of this.
Yeah, so that's the reason. So like now it's coming from SH or SD.
Well, we do these reactions competitively, separate vials. Okay, so we basically make this pyridinium from deuterated.
Separate.
Yeah.
Okay, I thought that
this isn't
taking SH and SD, then you will make it.
Yeah, we do both competitive and parallel experiments. Okay, so we do one where everything is deuterated. One reaction where everything's deuterated, we measure the kinetics. Another one where everything's protonated, we measure the kinetics, and we can compare those. We can also do an experiment where you have 50% of the thiol deuterated and 50% of this deuterated. And again, we see in the competitive experiment a 5-to-1 ratio.
Yeah. Assuming it's like let's say if you are having SH or SD, and if I am having PD, so there are two possibilities. Like let's say you can consider your SD is transforming to ND, one step, and second step is your ND is transforming this hydrogen to your carbanionic species. So if we start with the SD, then how we can make a consolidated statement that this transfer is active the RD state, or your PSD transforming to your phonium. That I am not actually able to.
You're saying that
Yeah. So
you're saying formation of this, or you're saying
formation of this one, or you're transferring to that one.
So when we ran these experiments, we actually made this independently.
Separate.
Yeah, we made this independently and subjected that either, again, the NH or the ND. We can make them separately and then use them as reagents, basically.
So this is now you're telling that breaking of this ND is the RD step, not the CD formation.
Right. Right. That's our interpretation. Which one?
The consent study of the certain state body, the way you have written,
the way we're showing it is concerted, and we don't necessarily have excellent evidence for that.
Charge as a Benjamin Jason, making from the calculus of that means you have very definitely all these groups atoms.
Yes.
But value is very less.
Yeah, so it's not a full anion. It's not a full minus. It's delta minus. Yeah, it's partial anionic charge. Yep, you're right. So you can see some of the substrates that we had here, and we could pick some of those out. I don't remember exactly which ones we chose for the Hammett study, but a variety of things with known sigma values could be pulled from our table. We prepared them, the substrates already, so we could easily pull them out of the refrigerator and use them for kinetic studies. And these are generally really easy to observe by fluorine NMR, right? So.
Okay, one slide, maybe you had shown that we're changing the pK to a
yeah, different mechanisms are probably occurring.
Yeah.
Yeah. So again, the initial hypothesis for the early work involved a deprotonation and the establishment of some type of an equilibrium and a low concentration of this as a nucleophile. Okay, and right, so if you look at the pKa's of TMG with the thiophenols, they lie in the appropriate direction. Yep, and same thing about the phenols with the TBD. Right, the equilibrium lies in that forward direction. That doesn't work here, right? Okay. I think you have to try things, right? And I don't think it's one of those things where some of the early amine bases we were using weren't working, so let's just try other things. And oh, pyridine's a base, right? And you throw it in there, not quite realizing that it's not going to work. It might work, but not because of what you think it or why you think it's going to work, right? So, but we learn and discover new chemistry, and we get to move forward. Yeah. Were you about to ask something or no? Okay.
Just before this slide, there is a yeah, that was here an ion product has a profile on the register group as well as the one for position. That is not.
Yeah, so it does have an opportunity to attack this position here. Again, that would be the other reactivity that we were talking about early on, right? The resonance effect activating alkenes. I speculate that it does occur, but it's reversible. So if you do addition here and you generate an anion that can then push back off the thiolate and allow it to attack back on the fluorinated position. So I think it attacks here, but I think it's reversible.
There is level one is constant. Now, then level one. So that is not involved in the.
No, yeah, at the end of these reactions, the alkene is actually in a stable position here. In most cases, it's walked into conjugation with the arrow ring or a hetero arrow ring. Okay, in a couple of cases, it's walked into a position that's kind of stabilized and protected, right? This is a hindered position here. Right, so they don't seem to further react under these reaction conditions to a great extent. Oh, yep.
Oxidation of UNCTD has some oxidation. So there is a potential for determinate elimination. It can make the molecule.
So you're talking about this? Yeah, so right. So you're thinking about this here, right? And so if we take these types of products and heat them up, they will do an elimination. And that actually makes the fluorinated alkene again, right? So this oxygen reaches over, deprotonates there. Electrons come down and they push off that. So yeah, if you take these sulfoxides, you heat them up, that happens. And so that's one of the processes that we think was occurring in some of the reactions as we were developing them, right? Some of them were under air. You could be decomposing by that mechanism.
Up to more than 100 you can.
Yep. 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 Yeah, if you have this product and you heat it up, that could occur. But under, remember when these are fluorinated, they're not prone to oxidize. So they don't oxidize with O2. the way normal thiols do. Okay, so with O2, this actually doesn't really happen. Right, so it's cool. It's cool to me. Sorry? Yeah, so for the fluorinated ones, you need a stronger oxidant here, such as MCPPA, right? That maybe there's some in between these two values, but up to peroxide won't touch these at 40 degrees in ethanol. So they're pretty stable.
Like if there is a chance of beta fluoride and beta hydride, I think beta hydride is more preferred.
Yeah, so beta hydride is preferred.
It is fluoride elimination should be also more equally preferred because the anion can stabilize by fluoride. Would be
The driving force for beta hydride as opposed to beta fluoride elimination? I know something that I'm not allowed to say. Not because of me, but email me that question in 2 months and I'll answer it. I'm not being protective of my own stuff, but
it's unusual, like showing the palladium 3 interface.
yeah, palladium 3. Yeah, that's weird. Do you want the full cycle?
So this chain walk with palladium is common, or it's a driving force of conjugation is driving force in this walk, chain walk?
Oh, I mean the chain walking can occur. So if you had a simple alkyl, if this was just an alkyl, those can scramble and leave the alkene at a lot of different positions. And so you get a mixture. There's no strong thermodynamic driving force.
No, my question
So-
like palladium also is common chain walk.
Oh yeah, palladium can do a lot of chain walking.
Nickel was more popular.
A lot of people are getting excited about nickel now, but palladium has a long history of these processes. Yeah, of doing these beta hydride eliminations, reinsertions, and chain walking to a stabilized position. Yeah.
So my last question.
I was gonna say.
Here you use specifically carbene ligand and also sulfonyl chloride.
Mhm.
So is there any importance for these two things? Because why not we can use iron bromide?
Yeah. So I'm gonna come back to that. I was just gonna say, look at this one here. If you think about this substrate, the alkene is initially here, and it chain walks all the way down to there, right? So you can walk that thing all the way down. The yield is kind of modest, but there's some oxidative decomposition of this coumarin. It's not the most stable. So your question again was about the carbene ligand?
Yeah.
Okay.
Is there any effect?
The carbene ligand wasn't essential. It improved the yield a little bit. It avoided some formation of biaryl. It wasn't essential, but it bumped the yield 10 to 15% in some of the early cases we were exploring. So we stayed with it, but it's not like it doesn't have to be there. Okay. The aryl sulfonyl chlorides are kind of interesting. I never would have.
Is it helping with chain walking?
Oh, I don't know if that's helping in chain walking. I'm not sure. Yeah. We observed the same products early on.
Because for
So-
me, I have not, maybe I missed palladium chain walking, not that many.
Yeah. In the last decade, Matt Sigman had a lot of examples of it.
Yeah.
There are plenty of others, but
Okay, you have proper.
yeah. Wait, right, his were palladium too, right? And so we might be a little bit different, but there are examples of that. Yeah, so I'll leave it at that. So Kedong, maybe one other comment, Kedong was the one who did this, and you overlap with him a little bit? No? He came just after you? Okay. And so Kedong, where's my arrow? Okay. He came from a graduate group that had used these aryl sulfonyl chlorides quite a bit and knew that they would make radicals. So he kind of entered this area without me encouraging him that direction for anything I thought. It was his previous experience that got us into these aryl sulfonyl chlorides. I probably would have started with an aryl bromide or triflate or something, right? So. No, we never observed those to the best of my knowledge. Yep. So you're saying, well, if the aryl SO2F, or sorry, if the intermediate from here reacted to make this bond, right? And we never observed that. I don't know why, or if Kedong observed it, I was never aware. But at 120 degrees, SO2 is very prone to leave. So I'm actually surprised that we did trap out this, as opposed to most of it making the aryl radical and then reacting. So that caught my surprise. He was able to characterize that.
So we are actually one accident. So see the screen that saying this. So we found a very good radical reaction. SOT is staying. SOT is not leaving.
It doesn't fall off with what?
It's leaving.
With what system or what?
Other customers like radical.
Does it? I mean, there's a transition metal in there.
No.
Okay, metal free. What temperature? 60 to 80. So if we're heating these up to 120, maybe the loss of NO2 is more rapid. I mean, it will be more rapid, but maybe that's enough to really push it over the to drive it off. I don't know. Okay.
No more question.
Yeah, so will it work with alpha methylstyrene? And so I don't think that's electrophilic enough, and I encourage you to try it for yourself and prove me wrong. Okay. You know, my gut instinct is that's insufficiently electrophilic if you only have one fluorine atom. The monofluorinated alkenes don't behave quite as, they're not as strong electrophiles. Okay. CF3 alkene. So you're saying if instead of this difluorinated, if I had a CF3 there? Here? If you had a CF3 there? Sorry? Say it again? Yeah, this was not very good at reactions in which this alpha position was substituted. Okay, and that's one of the additional reasons why we move forward here. And maybe one thing I didn't comment on here, but I should have, is that these fully substituted precursors were tolerated with the photoredox system where they weren't in the previous chemistry. So you're spot on your assessment. You know, the pyridine system didn't work if you had branching on that alkene. But now we can do it.
For coming here and giving very nice presentation. That's something of thanks, Professor Altman, for your department. Small token of
Thank you all. Good interaction.
It's our seminar in charge for Department of Medicine. So he will arrange all the He's supervisor.
And also, on photography.
Uh-huh.
And electrochemistry.
Do you have the icon plates? The photo cabinet, the electrochemical icon plates?
Like this one.
But he's a mix it down. He works on bio spectroscopy. Chinmay Kumar.
Mostly I'm working mostly catalysis was recently.
Which analysis?
Homogeneous catalysis. Recently I have started interested in this.
Sure. Which do you like more? Which do you like more?
Yeah. Mostly homogeneous because my background is from catalysis.
Sure. Yeah. Stay in chat. Yeah, thank you all.